my privilege to introduce today's keynote speaker, Dr. Matthew Harms. Dr. Harms is Associate Professor of Neurology at Columbia University and serves as at MDA's ALS Care Center, also at Columbia University. He directs an international multi-site effort using whole genome and transcriptome sequencing to bring precision medicine to ALS. His laboratory efforts occur in both the Motor Neuron Center and the Institute for Genomic Medicine. Dr. Harms received MDA's Diamond Award for his work directing an international effort with whole genome and transcriptome sequencing to bring precision medicine to ALS treatments. Dr. Harms also serves as a medical expert for MDA on ALS and other neuromuscular diseases. So it's been a privilege to work with Dr. Harms these last few months to plan today's symposium, and we are grateful for his willingness to chair the program today. And with that, we will turn it over to you, Dr. Harms.
Where'd your presentation go? That's right on the screen. So that substantially younger photo of me is a great place to start, actually. All right. Because we're a small enough group, and I feel like you should always know the people who you're getting information from. So I did want to share just a bit of my story, because I think it sets the framework for why we would bring spinal muscular atrophy and ALS together into a single Engage symposia. It goes back to when I actually looked that young with something that I would call a medical school mistake. So many of you probably don't know this, but you have to decide what residency you're going to pursue after medical school before you've even had a chance to survey the entire landscape of medicine. And if my pediatrics rotation had taken place a little bit earlier and I'd learned about pediatric neuromuscular medicine, I'd be standing here as a pediatrician today instead of as an adult neurologist, because I am secretly a pediatric neuromuscular specialist trapped in an adult neuromuscular specialist's body. I love taking care of kids, and that sets the importance of this next point, which is that I was able during my neuromuscular fellowship to redeem that early error by training at Washington University, where the neuromuscular fellowship included seeing and taking care of children with the whole range of neuromuscular diseases. And so since 2007, I've been in a muscular dystrophy care center, taking care of both pediatric and adult neuromuscular patients. My good fortune continued as I became a faculty member at Washington University, because my very first efforts at whole exome sequencing, as it had just come on, were to discover both an ALS gene and a spinal muscular atrophy gene, still continuing to unite the adult and pediatric side of what I was interested in. And that set the stage for me to move to the Motor Neuron Center at Columbia University, which is really a fantastic home, because this is one of the unique research institutions that looks at motor neuron disease health across the entire spectrum, from what makes a motor neuron, how does it develop, how do we keep it healthy, how does it deteriorate in spinal muscular atrophy, how does it hold on and then deteriorate later on in life in ALS. And that's also set me up to be able to pursue what I love, both the kids and the adults with neuromuscular disease, particularly motor neuron diseases, as I see patients in the Columbia ALS Center, the Spinal Muscular Atrophy Clinic, and then both the pediatric and adult muscular dystrophy care centers. So in many ways, I'm the appropriate person to try to bring together spinal muscular atrophy and ALS, these two different ends of the spectrum, for today's symposia. Other Engage symposia will be taking place across the country over the years that will pair other diseases that are covered by the Muscular Dystrophy Association. And we're getting to kick it off here with the return to in-person Engage symposia with this particular motor neuron-focused effort. I also, because of my diverse background in neuromuscular disease and my focus on both kids and adults, I've joined as a strategic medical advisor to the Muscular Dystrophy Association, which has been a tremendous pleasure, allowing me in yet another facet of my life to keep my foot in both the pediatric and adult worlds. So I'm extremely privileged and extremely lucky to be in that circumstance. And I also think it gives me an opportunity just to speak about the advantages of an organization like the Muscular Dystrophy Association. I think it's important, and there are roles for the individual nonprofits and organizations that represent each of the individual diseases, like ALS or spinal muscular atrophy or the different types of muscular dystrophy. But there are also some major advantages to having an organization like the Muscular Dystrophy Association that is a big tent for all of them, and I think I've outlined them here. One, it provides that common voice for advocacy to make sure that all of our shared concerns are being brought before legislatures as well as insurance companies. Obviously, there's a very strong overlapping clinical need and clinical expertise, as my own career manifests. There are also many shared disease mechanisms, some of which I'll touch on in the next 30 minutes or so. And I'd like to point out, and I think many of the things that I'll talk about today will emphasize how research in one neuromuscular disease can immediately translate into benefits and understanding for other neuromuscular diseases. We're learning more and more by breaking down silos between these different neuromuscular disorders, and this is especially relevant for the development of therapy. So, like I said, this Engage Symposia is mostly focused around one common theme, one common type of cells in our bodies, and those are the motor neurons. So we're learning through efforts called single cell sequencing that there are literally thousands of different types of motor neurons, but they all share some common traits. We kind of parse them into two groups: the upper motor neurons, which are the ones living in the brain, which are in the region that's labeled, colored in red here. And then the lower motor neurons, which live in the brainstem and the spinal cord, which are colored in green in this diagram. And if you zoom in to kind of get a look at what some of these motor neurons look like, the blue dot there in the spinal cord is the lower motor neuron, and it sends something called an axon all the way out the peripheral nerve. Let me actually see if I can get my highlighter here. Yeah, I think you can probably see it. Sends it out the peripheral nerve all the way to the muscle, where it latches onto the muscle and then instructs the muscle what to do. So the upper motor neurons here send the signal that it's time to move. Those signals travel all the way down to the lower motor neurons, and then the lower motor neuron here is stimulated and it tells the muscle to contract, and that's how we get strength. And these neurons are actually born around 3 weeks of gestation, so while the embryo is still the size of a poppy seed. And these motor neurons basically are born and immediately connect to the muscles that they're right next to in that tiny little embryo. And then over the course of our lives, as the embryo grows, and then as we grow as humans, that little wire connecting the motor neuron to the muscle gets longer and longer and longer and longer, such that by the time you're 6'2", like me, in your lumbar spine, one of those little wires extends all the way from about the level of my ribs down to my big toe. So almost a full meter in length. And of course that means that a motor neuron is extremely complicated. It has to have the nucleus with its DNA information all the way up in my spinal cord, but still control things that are happening a meter away at its connection to the muscle. And this, of course, makes motor neurons some of the most complex and wonderful and amazing cells in the entire human body. Now, there are other doctors that will argue that their cell type is the most interesting, but I think we'll win. So the diseases that affect the motor neuron are roughly parsed into these categories: spinal muscular atrophy, which is caused by mutations in the SMN1 gene, amyotrophic lateral sclerosis, and then there are other diseases, other types of spinal muscular atrophy which are not caused by SMN mutations, and then another disorder called spinal bulbar muscular atrophy, or Kennedy's disease. And I would like to point out that research into these diseases has really taken off over the course of the last 2 decades. Here I've plotted all of the research papers for ALS and spinal muscular atrophy as they've accumulated since the 1870s, which is about the time that both of these disorders were described. And I hope you'll appreciate that really over the last decade, the number of scientists that are devoted to finding new aspects of why the disease occurs, what we could do about it, has really skyrocketed, such that now there are hundreds of papers being published on each of these diseases each year. When I talk to patients about why this has me optimistic, I oftentimes use an analogy to self-driving automobiles. Up until 5 years ago, the technologies to make something like a modern Tesla work and work reliably just weren't there. We didn't have battery life that could support the long-distance driving. We didn't have GPS that was accurate enough to within meters to know exactly where cars were. We didn't have digital processing of camera footage such that a computer could take in the environment through its cameras, translate it into whether it was a person or a dog or a bike or a lane marking. And all of those technologies had to coalesce at the same time in order to make something like a Tesla possible. By investing in battery technology and GPS and faster processors and higher resolution cameras and AI technologies and things like that, simultaneously, the number of technological developments took place such that kind of all of those came together at the same time to be able to produce automatically self-driving cars. And what I think this illustrates, what this diagram illustrates with the massive ramp up in research publications and information that's available for each of these diseases, is that we are just approaching that threshold where our ability to understand biomarkers and to do good clinical trials and to have therapeutic targets for which we can develop drugs, the chemical developments that take place to modify the medications that we're testing, all of these things are almost at that threshold where one day, maybe next year, maybe the following year, maybe the following year, all of them will coalesce, come together, and we'll have essentially the equivalent of the Tesla for neuromuscular diseases in general, and ALS and SMA in particular. We're already starting to see some of the fruits of those efforts. Nusinersen was approved in 2016, and gene therapy for SMA in 2019. And just almost exactly a month ago, an antisense oligonucleotide for one form of ALS was approved called tofersen. So because we have people in the audience today who know a lot about SMA because it's affected their family, or know a lot about ALS because it's affected their family, but maybe not both, I thought I would take a minute that because these two diseases both now have FDA-approved molecular therapies linking them, to introduce each of you to the other sides, the other group of people's disease. And I'll do that by starting with spinal muscular atrophy because it's the one that had a molecular therapy approved first. So spinal muscular atrophy is known by many historical different names. There are multiple types that we see in the clinic. So the most tragic type is type 0, where babies who have this usually are stillborn because they lack any ability to move at all because their motor neurons are essentially non-existent. And then the severity spans all the way up to individuals who, as adults, begin to notice proximal muscle weakness of unexplained cause, and they too have spinal muscular atrophy. The most common form is the one that's called type 1, and these babies used to, somewhere between 0 and 6 months of age, start to lose the ability to move their muscles, to lift their heads. And in an illustration of what is now quite a rare outcome, you can see the baby illustrated here doesn't have really any muscle strength left. Eventually, these babies would need to have a tracheostomy to help them breathe, need to have a feeding tube because they didn't have a strong enough swallow reflex in order to take in enough nutrition, and would be very debilitated and oftentimes pass away before the age of 2 because of complications of being unable to breathe or move. But despite the severity range of never being able to move to being able to move until late in adulthood, we now understand that spinal muscular atrophy has a shared genetic cause. In all of these individuals, no matter the severity, they are missing or have defective copies of the SMN1 gene. So by the end of the next 10 minutes, you're going to be a molecular biologist, if you aren't already. But I promise you'll get through this. Hopefully it's not bringing back PTSD or something from your middle school or high school biology days. So this is an illustration of the SMN1 gene that is the cause of spinal muscular atrophy. Every human, or almost every human, has two copies, one that they inherited from one parent and the other copy that they inherited from the other parent, and the gene itself is illustrated here in red. Okay. There we go. Okay, it's working again. So this is the DNA level. This is coiled up in the nucleus of each of your cells. And when the SMN1 gene needs to get utilized, because at the very end of this you want to have SMN protein, the cell turns on the DNA and copies it into something called RNA. And what I'd like you to notice is that when it copies it into RNA, it's actually in multiple little tiny pieces that are distributed across the DNA. These are called exons. And before the exons can be utilized, they have to be stitched together in something called splicing. Remember that because it's going to come up again later. So you go from multiple little pieces, all of them need to be put back together in the right order and with all of the pieces in place in order for the body to then use the RNA to do something called translation, which takes the code in the RNA and turns it into the protein, in this case, the SMN1 protein. So in the normal situation, those of us who have two normal copies of SMN1, we make 100% normal RNA, which then gets translated 100% into normal SMN protein, and our motor neurons are happy. So then, if you take the parent of a typical child with SMA, they're usually missing one copy of the SMN gene. So they've got the SMN1 gene here, but on the other chromosome, the copy that they inherited from one of their parents, they're actually missing it. So when the body needs to use the DNA to make more SMN protein, it turns it on, but it can only make half as much of the RNA because it's missing a copy. And so carriers who don't get any motor neuron disease have 50% of the amount of the SMN1 protein. And that's enough. Motor neurons are happy and perfectly content to live life with 1/2 of the amount of protein. But then, if somebody who is missing one copy has a child with another person who's missing a copy, and that child does not inherit a copy, and so missing both copies, then they will make 0 RNA and 0 SMN1 protein coming from this particular region of the genome. And that's how someone develops SMA. They don't have enough of the SMN1 protein to keep motor neurons alive. There are some rare cases where they're missing one copy, but the other copy has a mutation, so it doesn't work either. And so you end up with less SMN1 protein than you need as well. So thankfully, the human evolution has led to the creation of a backup copy of SMN1. Unfortunately, it's a defective copy, and so it's only partially effective. So here's that normal copy of SMN1. And literally at one little tiny location, there's a C, and in the backup copy, there's a T. And that backup copy T has a dramatic effect on the body's ability to use it. So if you have the C there in the normal copy of SMN1, all of those little pieces of the gene at the RNA level can get stitched back together in the normal fashion. And when stitched back together in the normal fashion, gives rise to the normal protein. If you have a T there in that backup copy, which everyone does, no one has a backup copy without that T, it's just the rule, it leads to the splicing going wrong. So instead of stitching together all of the pieces in the right order, it skips piece 7. And when the RNA is missing piece 7, it leads to an abnormal protein that gets degraded very quickly. And so you don't make enough SMN protein from the SMN2 backup gene. Now, the cell isn't perfect, and so it makes a mistake every once in a while, and thankfully it does. Because in this case, when it makes a mistake, even though it's supposed to skip the 7th piece because of that abnormality, it forgets about it and it ignores it, and it does go ahead and include the 7th piece. So on rare occasions when the body is using the backup gene, it actually makes the normal protein by accident, essentially. And that's fortuitous because that means that 15% of the time, you can generate a little of the normal protein from the backup gene. So it turns out that the number of backup genes that you have is a major player in how severe your disease and how early you get the symptoms. So if you have one copy or fewer, some people have 0 copies of the backup gene, that's when you get that really severe situation where the baby in utero doesn't move and is usually stillborn. And then the more copies of the SMN2 backup gene you have, the more accidental protein you're going to make, the less your motor neurons are going to be irritated and the longer they're going to live. And so you can see that with increasing copies of the backup copy, you have later and later onset and milder disease. So that led researchers from the very beginning of figuring out how all of this was working to the hypothesis, which I think is now proven, that you could fix this by replacing the normal copies of the SMN1 gene. If you could increase the amount of times that the body used this, you would increase the number of times that it would make a mistake and make the normal protein. And so you could drive the gene use of SMN2 and make more SMN1. Or if you could correct this missplicing and make it always include exon 7 or usually include exon 7, you could also then end up with more SMN protein. And 2 of these 3 strategies have been quite successful. The first of these I already mentioned is that using an antisense oligonucleotide called nusinersen, you can restore the splicing and get more SMN protein. A small molecule called risdiplam can do something similar. And then there's also a gene therapy where you can deliver the SMN1 protein gene for the protein so that you can restore the expression. And both of these have dramatic effects, or all 3 of these 2 approaches have dramatic effects on the longevity and strength acquired by children with spinal muscular atrophy. Now in New York State, where we have prenatal screening or neonatal screening for SMA, children are getting treated with the gene therapy and then one of these other molecules afterwards, even before they have symptoms. And that is saving neurons and leading to nearly normal motor development in some cases. So a dramatic turn of events over the last 5 years for children that usually had passed away by the age of 2 are now attending school, are now getting by with much less weakness than they would have had, and which is a tremendous, nearly miraculous event. So I just wanted to pause and talk a little bit about gene therapy. So onasemnogene, which delivers the SMN1 protein into the neurons, is one of the therapies that we use as early as possible in children with spinal muscular atrophy. But it's not the only way to deliver a gene, and I just want to highlight that researchers are working very hard to be able to deliver gene therapy using a whole range of techniques. The viral vector that's used to deliver onasemnogene is just one of those, and I think over the next years we'll see other and increasing innovative ways of delivering genes into different neurons, or cells for that matter. So while this seems like a miraculous cure for neuromuscular disease, there are some challenges that we're all working on. And these are kind of those threshold challenges that I'll mention, kind of like getting to the Tesla. Like, we have to be working on all of these and bringing them all across the finish line in order to make big changes or big advances. So the first of these challenges is that many of us are walking around already with immunity to some of the viruses that are currently being used for gene therapy. That's because viruses are not that clever. They're very similar one to another. And so when you develop an antibody, for example, to one coronavirus, sometimes it'll keep out another coronavirus, even though they're different. And so many people are walking around with immunity already because of exposure to one virus that would block a gene therapy from being used for their disease. Once you've been given a gene therapy, then your body definitely makes antibodies and immune response that would prevent it from being used again. So that means that gene therapy, as we currently have it, can only be given once in general. And then after that, your body would neutralize it and prevent it from getting into cells and would keep it from being effective for your disease. Another problem related to the immune system is that some people, when treated with gene therapy, have a very strong immune response. And that strong immune response can cause liver failure, it can cause inflammation in the muscles or within the heart itself, and in some circumstances and in some diseases, it has actually led to deaths. So it's not anything to mess around with and something we have to take very seriously as we think about developing gene therapies for a whole host and range of neuromuscular diseases. Another challenge is that it's hard to manufacture large quantities, such as what would be required to treat an adult. Right now, the SMA gene therapy is approved for use in infants who are quite small. And so you can make enough in order to deliver it. But as we envision treating later onset diseases and larger individuals, this will be a challenge. And then while the neurons that SMA where we think that the virus is having or the gene therapy is having its biggest effect in SMA, those neurons don't divide. And so when you deliver a copy of the gene, it stays there and continues to make SMN protein. But in other diseases, like muscular dystrophy, the cells continue to divide, and the gene therapy doesn't. And so as muscle fiber, for example, that got treated with muscular dystrophy gene therapy divides into two, only one of those muscle fibers will have the gene therapy, and the other one will go back to having no gene therapy and it being as if it wasn't treated. And eventually, over time, we expect that the effect of the gene therapy will wear off. Another challenge is that gene therapy can only deliver small genes at this point. And so if you have a very large gene, you have to find out if you can reduce it down into a mini gene. Can you figure out which parts of the genes are important, stitch them together, and make a little mini version of the gene and deliver that? Though there are some genes in neuromuscular disease that are small enough to consider putting in by themselves, like the SMN1 protein, most of them are too large, and so people are coming up with strategies for ways to stitch them together, to give two viruses, one with half of the gene, and a way for the two genes to come back together at a later point inside of the cell. And then finally, it's important to know that once you put in a gene therapy, it's going to be turned on and keep manufacturing the protein product until the DNA gets degraded or until the cell dies. And that means that it's hard to turn down the protein product. And so there are some diseases where we know if you give too much of the protein, it's actually toxic to cells. And so we need to develop additional ways so that you could make a gene therapy product that only turns on a little of the protein, or that you could turn it on and off with other medications or something along those lines. So where are gene therapies being used in neuromuscular disease? Well, so there's spinal muscular atrophy. I won't have time to talk about it today, but there's one gene therapy that's that's on the drawing board for ALS, the other end of the motor neuron spectrum. But it's up for FDA approval for Duchenne muscular dystrophy, and next month we should hear from the FDA as to whether that's going to be approved. And then it's under investigation for X-linked myotubular myopathy, as well as some limb-girdle muscular dystrophies. Now, if you would like to really geek out on gene therapy, one of the other Engage events that's being hosted this month is Gene Therapy 101, and it's being given by my very good friend Craig Zaidman and one of the nurse practitioners at WashU on Thursday, June 15th from 4 to 5:30. This is an online event that you could register for, and it's also available in Spanish if that's easier for you to understand. So look on their website if you're interested in learning more about gene therapy, in particular for neuromuscular disease. Now, the other FDA-approved medication for spinal muscular atrophy that I'd like to focus on because of its relevance to ALS is the concept of antisense oligonucleotides. So no one likes saying that long word, so we just call them ASOs, and you probably have heard people referring to them by that name. And ASOs are little synthetic pieces of DNA. You can think of them that way. It's not exactly right, but that's essentially what they are. And they are designed to actually recognize the RNA, the part that gets spliced that I was showing you earlier, in order to manipulate that RNA. And it can do so by a number of different mechanisms. I'll focus on these top two. The middle one here, the antisense oligonucleotide shown here in blue, actually sticks to a part of the RNA that controls how it gets stitched together, how that splicing takes place. And it can actually influence it enough to either force it back to the normal splicing or to force a completely different type of splicing. And that's exactly how nusinersen works for SMA. It helps the body include exon 7 that would normally get spliced out. It now retains exon 7 to become the normal protein. In Duchenne muscular dystrophy, it actually induces the skipping of an exon. So instead of convincing the body to include a chunk of DNA, it's actually convincing the body to excise and get rid of a piece of RNA that you want to skip over for purposes I won't go into today. The other way that you can use antisense oligonucleotides is actually to force the body to degrade the RNA. So let's say you have a gene that's overactive or a copy of the gene that you think is doing damage. You could manufacture one of these antisense oligonucleotides to stick to the RNA, but instead of convincing the body to do splicing differently, it actually convinces the body that it's a virus attacking the body or attacking the cell, and activates machinery that's designed to chew up viral DNA once it gets into our cells. So when a virus infects us, gets into the cell, the body is looking for abnormal pieces of DNA that it doesn't recognize, and when it finds them, it chews them up to try to prevent the virus from giving us all a disease, right? So in this case, we're harnessing that by giving an antisense oligonucleotide that sticks to the RNA, and then the cell interprets that as being a deadly virus it needs to get rid of, and so it chews it up, and now you don't have the RNA. And if you recall in that diagram, the RNA is necessary to be translated into the protein, and so now you can't manufacture the protein that is coming from that gene. So it gives you a way of turning off the protein, or turning off the gene that you think is causing the problems. And that's where we're making a lot of progress in ALS, and where the recent approval of tofersen comes in. So we're all very excited that the FDA approved Qalsody, which is an antisense oligonucleotide directed against the SOD1 gene, which I'll talk about here in just a moment. It was approved under an accelerated approval mechanism, which is unique for the FDA. It doesn't do that very often, but did in this circumstance, which was quite great. And basically, this is how it works. Like what I was saying is if you've got this ALS-linked gene mutation, the mutated DNA leads to mutated RNA, and when you try to use the mutated RNA to turn it into protein, it becomes an abnormal protein that's toxic in this case. So now when you have this ASO, that's it right here in this little black, it recognizes the mutated RNA, tells the cell to destroy it, and now the RNA is not present, and so no protein can be manufactured. So SMA and ALS both have approved antisense oligonucleotides for their therapy. That's one link. And one other link I would like to point out is that because nusinersen had gone before, and we had learned from our experience with SMA how to efficiently roll it out, how to get it into our pharmacies, how to get it to patients, and had done all of that a few years prior for SMA, we are now much more efficiently rolling out tofersen for patients with SOD1 as a result. So that's another way that having organizations like the MDA that keep ALS researchers and ALS clinicians in the same room with SMA clinicians and SMA researchers benefits us all, because we learn from one another's experiences. Those who are on the vanguard and leading can turn around and inform what happens next in another neuromuscular disease. So with the approval of Qalsody for SOD1 mutation ALS, that gives me a good opportunity to pivot now and talk a little bit about ALS so that the people with more experience with spinal muscular atrophy know a little bit more about the disease. And I'd just like to commemorate that yesterday was Lou Gehrig Day, because on this day in 1941 he passed away from the disease. So Major League Baseball at all of its stadiums was raising money to help ALS. The clinical signs of ALS are usually stiffness, spasticity, and muscle wasting. I won't go into the details here, but here in the upper box is one of the hands of someone who has ALS, where the muscle has atrophied away. And though we don't do this very often, when you look in the spinal cord, you can see scar tissue, speaking to the lateral sclerosis part of the name. So what are the symptoms of ALS? Well, they're really a constellation that varies from person to person. So they're all caused by progressive neuronal degeneration. The upper motor neurons, again highlighted in red like the first time I showed you the diagram, give rise to pseudobulbar affect, where people have abnormal laughing or crying attacks. Spasticity, where the limbs are very stiff and hard to move quickly. And then when they come to clinic, we see hyperreflexia, where you hit a reflex and it's much, much larger and far brisker than it's supposed to be. In green are those lower motor neurons, and just like in spinal muscular atrophy, when those deteriorate, you get profound weakness, muscle fasciculations, and eventually atrophy of the muscles. We're also learning that in ALS, non-motor neurons can also be affected. The motor neurons are the most susceptible and are hit first and earliest and hardest. But in some people, non-motor neurons, including the ones shown in blue, which give rise to what we call executive function, can also be affected and lead to additional symptoms, including dementia. Now, in clinic, SMA looks the same across people. It's always those lower motor neurons that deteriorate. And so it's the atrophy, the fasciculations, and the weakness that we see. But in ALS patients, the disease can look very, very different across, take 100 ALS patients and no two of them are going to look the same, because there's always a mixture of these upper, lower, and non-motor neurons that are involved, and they vary from person to person. Some people only have upper motor neuron involvement. We call that primary lateral sclerosis, and that takes place in 5% or so. At the other end of the spectrum is progressive muscular atrophy, where only lower motor neurons are involved and look much more like the spinal muscular atrophy cases. So ALS is about as common here in the United States as spinal muscular atrophy. So it's a rarer disease than spinal muscular atrophy. But the best data we have for how likely someone is to get it comes from studies of white men. This is changing, thankfully. People are now going back and taking the time and energy to look at it in other patient populations, as should have been done decades ago. But a white male actually does end up having the highest risk of developing ALS, 1 in 370, which means one of the people I graduated from high school with is likely to get and have ALS. That means that in the New York metro area, there are more than 1,000 patients, and worldwide, more than 300,000. 10% have a family history of ALS, but 90% do not. And even though 10% have a family history, we still find identifiable gene mutations in 15%. And by studying the families, we've actually discovered quite a few ALS genes. So SMA, 1 gene. ALS, the opposite end of the spectrum, somewhere between 2 and 3 dozen genes. We're still working out the details of all of the ones that are coded here in yellow. The ones that are in green are ones that we're fairly confident cause ALS. So with this many genes, we can now go and do large-scale sequencing studies in patients with ALS to find out what proportion of patients with ALS have these gene mutations. So shown here in the pie chart is a representation of that. So the vast majority of times when we find gene mutations, it's in the C9orf72 gene, followed by the SOD1 gene. That's the one that tofersen is now approved for the treatment of. And then each of the other genes is increasingly rare from that point on. All told, if you don't pay any attention to family history, 11% of people will have one of these gene mutations. Now, I want to emphasize that these numbers come from European studies. We're just getting started on studying other populations, but we already know that outside of the European population, the rate of that C9orf72 gene is lower, though it's still detectable. And in fact, there's a higher rate of TDP43 and FUS gene mutations in those other populations. So one of these common genes we now have an FDA-approved medication for. The ones shown here in yellow have ongoing clinical trials of antisense oligonucleotides, what I showed you, the attack the gene, knock it down so that the body can't manufacture the protein type of strategy. And then in a program that we're running at Columbia University called Silence ALS, we're actually in the process of developing antisense oligonucleotides for even rarer versions of these rarer genes that cause disease less frequently. We have things in development for TAR-DBP, as well as the ones that I've highlighted over here on the right. So with an approved medication, several others in clinical trials, and the opportunity to create antisense oligonucleotide therapies for many of the rarer forms, we have now created ALS genetic testing guidelines. And I may have parodied a famous movie's title for my motto for genetic testing, and that is everything, everyone, all at once. So that's because 15% of people have an identifiable cause. 60% of those have a family history, 10% do not. And it's really hard for us in clinic to predict which gene might be mutated, so we really do need to sequence all of them at one time. Furthermore, about 2% of the time we actually find 2 different mutations that could be playing a role in an individual. And so if you only look at one of the genes, you're going to miss the other possible contributors. And I think the important point is that by doing genetic sequencing very early in the course of diagnosis, we'll be able to direct people either to treatment with tofersen if they have an SOD1 mutation, or to clinical trials for the specific gene that they do have mutated, if in fact one is found. The good news is the genetic testing is currently free at a couple of locations. I won't go into that. And even if those programs were to go away, most insurance companies have now gotten on board with the idea that genetic testing plays an important role in the diagnosis of ALS. So just to put this in kind of a visual diagram, so this is 100 people in an ALS clinic. This top row represents the number of individuals in which we can find a family history of ALS or frontotemporal dementia. But that means that the rest are what we call sporadic ALS or singleton ALS. But now if we offer sequencing and get sequencing on everyone in our clinic, this is what we'll find. That in that top row, 70% of individuals will find the culprit mutation and know what to do next. But if we sequence everybody else, we'll find even more individuals who carry those same gene mutations that had no idea that they were at risk for carrying one of those gene mutations and may not have known that they were eligible for clinical trial or for treatment with tofersen. I will just point out that, in family ALS is where we've done all of our really successful gene discovery, but we still don't have answers for 30% of people with family history of ALS. And that, I think, tells us that more genes remain to be discovered. And people have said, look, you've already discovered 24. Why do you need to keep on discovering new ALS genes? Don't we know enough from that? I think the counter argument to that is that we now have the technologies to develop those antisense oligonucleotides for the rarer genes. But also with each gene that we discover, we're learning about new pathways that are important in ALS. So going back to this diagram, when we learned about senataxin, we learned that DNA coiling was important. When we found TDP43, we learned about RNA processing. When we found ubiquitin-2, we zeroed in on autophagy as a pathway. Basically, each one of the ones that I've circled here has opened up a new avenue of discovery and investigation in ALS by giving us a genetic clue to a new problem within the cell that could lead to the disease. So I liken this to climbing the world's tallest climbing wall. I'm not interested in doing this. This looks harrowing, if you ask my opinion. But with each of these genes that we discover, we're ascending the wall of total, and complete understanding of the ALS disease mechanisms. And we can start to implicate different driver mechanisms within ALS. So by taking the genes that we've discovered and their functions, and then grouping them, we see that the ability of a cell to control protein quality and to get rid of proteins when they've outlived their usefulness and replace them if they're dysfunctional or misfolded is important. The ability to ship molecules all the way from the cell body in the spinal cord all the way out to the muscle in my toe is very important, and we call that cytoskeleton and axonal transport. And some other mechanisms are also of key importance. And so as we discover more genes, we're also going to be discovering more and more of these pathways. The good news is we're doing that. In 2015, when I first came to Columbia, we did an analysis of 9,000 participants and identified 2 new ALS genes. We then did one with 11,000 participants and got clues to some additional ALS genes. Now in our current iteration, we're up to 27,000 participants. And we have in the works plans for 100,000 participants. And so with each of these different gene discovery efforts, we're finding new clues to what's causing ALS, and that makes me very optimistic. Even more optimistic is the fact that as we've done these studies, we've brought together people who never used to work together. So all of these organizations have now banded together and put all of their data into an organization called ALS Compute, which is going to have access to any researcher who can apply to get the data and analyze it. And we have collaborations with the European equivalent to co-analyze our data in order to discover more ALS genes. So let me just pause there and give the ASO summary for gene-positive ALS, what I've talked about so far. So currently we have an approval for SOD1-related ALS, that's tofersen or Qalsody. In clinical trials, we have ataxin-2, which is a study called ALSPIRE or ALS-SPIRE. For FUS, we have a study called FUSION. And then in the Silence ALS program and others like it, we have targets for TAR-DBP and other rare genes. So that's the good news side of the column. But with all research, there are challenges. And so on the negative side and on the downer side is the fact that some ASO studies have been either negative and did not show a result, showed that they might actually be worsening the disease a little bit, and have led to either the studies being halted or the drugs discontinued. And this week we just heard about another of the wave therapy against C9orf72 gene being unsuccessful. I've got lots of thoughts about why that's the case, and I don't think we have to give up yet. Every gene is turned on in 2 different directions, and our ASOs usually only target one direction, and I think we have to target both, or maybe the other direction. So I think there's still hope, but when you develop drugs, sometimes you're going to fail, and it's not always going to work. Other thing I'll point out is that in ALS, there are some mutations that are the wrong kind to attack with an ASO. Those genes include optineurin and TBK1 and other genes that are what we call loss of function, where you need to actually boost the gene as opposed to getting rid of it. And so those will require us to continue working. So that's all of the gene-positive individuals, but there are, as I told you, a lot more individuals with gene-negative ALS. And so I'd just like to conclude by showing you how we're thinking about using antisense oligonucleotide therapy to target all of the rest of ALS, which I think is extraordinarily exciting. So it begins with the observation when you look in the spinal cord and cortex of people who have died of ALS, that cells have these big clumps of brown goop sitting outside of their nuclei. I don't expect you to be able to histologically recognize that. That's a different class. I'll show you a diagram of what that looks like. So basically, if this is a cell, and in blue here is the nucleus, the protein called TDP-43 is supposed to be clumped up in the nucleus. It does sometimes go out into the cytoplasm, but for the most part, it's supposed to be inside the nucleus. That's the normal healthy neuron situation. When we look at an ALS, we see that TDP-43 has left the nucleus and is now clumped up in the cytoplasm. So how might that be leading to disease, and would that potentially be a target for antisense oligonucleotides? So very smart colleagues of mine have discovered that TDP-43 actually regulates how splicing takes place for a good number of genes in the human genome. So this is back to that diagram of the splicing for SMN1 and SMN2. Now, take this to be other genes. We don't know if this is happening for SMN1 and SMN2. But basically, in this circumstance, where TDP43 isn't in the nucleus to do its job, missplicing takes place and it goes wrong. So for example, a protein called UNC13A is essential for the connection between the motor neurons and muscle. And when TDP43 is there in the nucleus doing its job, you get normal splicing, which then leads rise to a normal amount of UNC13A protein. But if TDP43 is now out in the cytoplasm and not in the nucleus where it does its job, now you get missplicing, and just like with the bad copy of SMN2, you end up producing less of the normal protein, and so you don't have enough UNC13A, which contributes to the death of neurons in ALS. UNC13A is not the only protein. There are some 50. Another one that's receiving a lot of attention is one called STMN2 or STMN2. Same thing happens. When TDP43 is outside of the nucleus, missplicing takes place, and it actually includes a new chunk of DNA that's not supposed to be there. And that abnormal chunk of RNA leads to an abnormal protein that the body recognizes as abnormal and deletes it. So we now know that reduced STMN protein is seen in humans with ALS. We know that if you make this happen in mice, they do lose their motor neurons, so it's enough to kill motor neurons. And that in human cell models, it causes the neurons to be dysfunctional. So we think that this may actually be part of the disease process. So now, cryptic exon splicing block ASOs are being developed and in clinical trials to try to reverse this abnormality in STMN2, to bring up the levels of STMN2, and to bring up the levels of UNC13A, and probably many others of those 50 that I was mentioning. So it's also possible to use antisense oligonucleotides to try to force TDP43 back into the nucleus. I won't go into all the details, but ataxin-2 seems to promote the level of ataxin-2 protein in your body, seems to promote the movement of TDP43 out. And if you knock it down with an ASO, so directing against a completely different gene, you can theoretically drive it back into the nucleus. And that's another clinical trial that's ongoing. So the takeaway points from my talk today is that rapid advances in genetic therapies have really revolutionized the care of patients with SMA. Genetic therapy is just beginning for gene-positive ALS, and it's not going to be as transformational as in SMA, but we'll get there. Lessons that we've gleaned from the treatment of SMA and of genetic ALS are poised to impact even the gene-negative ALS cases, as I just was illustrating with ways to use ASOs even in people without identifiable gene mutations. And hopefully, as I've tried to bring together the SMA worlds and the ALS worlds, you'll agree with me that diverse neuromuscular diseases can really benefit from learning from one another, from cross-pollination, and from collaboration within an organization like the Muscular Dystrophy Association. And with that, I'll go ahead and stop.
I was wondering if you think your vision is that each different type of ALS, I know that you mentioned there's many different mechanisms, different cellular mechanisms affected by the different gene mutations. Can we go upstream enough to find a common, a mechanistic pathway to target? So one drug to treat all the ALSs, or is your vision that each one of the different mechanisms targeted will require a different medication?
Yeah, so when TDP43, basically the question is, can we go even upstream of the TDP43 leaving the nucleus, right? So that's seen in 90% or 95% of people with ALS. Can we figure out why the TDP43 left the nucleus, go upstream, find all of the different ways? I think we're going to find multiple ways to force TDP43 out. And so I think that no matter how proximal we go, we're going to find multiple triggers. And so we will need to be able to identify which patients are in each subgroup and then pick the right medication. That maybe it's an ASO, maybe it's a combination of ASOs, to tailor it to individuals.
Okay, sounds good. Do you have a question?
So, a great presentation. I have two questions. Sorry, they're going to be It's going to go in the weeds. So the first question is regarding the accumulations of the protein TDP. 43. Just learning from the Alzheimer's world and other aggregation, diseases. Does the field know for sure it's a cause and not a consequence of the disease? So, because you don't want to be just treating the consequence of a disease, and whether that is the root cause, because what if you get rid of the aggregations and it doesn't do anything because it was more consequence?
Yeah. So in particular for the clumps of the TDP-43 protein, we don't know how much of a role that's playing in causing neuronal degeneration, right? It might be a protective mechanism. Like it might be good for your TDP-43 to be clumped up in the cytoplasm rather than floating around doing whatever it does when it's not clumped up. There are 2 drugs that are about to enter clinical trials in the platform study whose goal is to prevent the aggregation of TDP-43 by an indirect pathway, but one nonetheless. And they actually have a really good way of telling in the spinal fluid whether that happened or not. So I think we're going to learn from these clinical trials whether disrupting TDP-43 clumping is a good idea or not.
Cool.
But you're right. We don't know exactly whether it's loss of the TDP-43 function or the clustering or clumping or both that's contributing.
Cool. And the second question is, I noticed from all your gene lists, because this is my area of research, that a lot of these genes also cause other forms of diseases, including neuromuscular diseases. And can you comment on, knowing that fact, how that could help you in strategizing therapeutic approaches to ALS? Are they different classes of mutations, missense versus nonsense? Are they different inheritance patterns on these genes versus what we see in ALS?
Yeah.
Or do they cluster in particular places of the gene because of gene function?
Yeah. So what he's referencing is that the blue dots on my gene graph also are genes that cause other neuromuscular diseases. And by and large, those are the same mutations that cause the other neuromuscular disease. It's probably just the case that in a handful of individuals, for example, VCP, which usually causes a muscle disease with dementia and sometimes bone involvement, that there's a subset of people who have more vulnerable neurons. And so they look more like ALS at the beginning of the disease or by the middle of the disease, but have features of both. So there aren't, with the exception of KIF5A, where there's mutations at one end versus mutations at the other end that stratify. Most of them, it's just how the patient looks in clinic, I think, that is the differentiating factor.
So for muscular dystrophy, do you have the same research going on and there's many different genes involved?
Yes. So yeah, there are a handful of dominant genes that cause the more common ones: facioscapulohumeral dystrophy, Duchenne muscular dystrophy, and then some of the more common limb-girdle muscular dystrophies. But yes, there are just as many genes, if not more, for some of the other neuromuscular diseases. So limb-girdle muscular dystrophy just had to completely redo its classification system because it ran out of letters. So there was like limb-girdle 2, 3, A, B, C, D, E, and once you run out of letters, and so they've revamped all of that. Charcot-Marie-Tooth neuropathy, which is another disease that's cared for by the Muscular Dystrophy Association, has close to 200 causative genes. So there are, one of the challenges of neuromuscular medicine is just how many different disease genes there are, but that also means that there are lots of opportunities for developing molecular therapies.
I'm just really curious about the familial forms that you can't find the root cause of the gene. Do you think it's a variant interpretation issue, or do you think it's occurring in regions of the genome maybe that's like really difficult to capture? Or Do you have any thoughts on that?
Yeah, so it's unlikely to be variant interpretation because all of these families, ALS geneticists want an explanation for these families. So any variant that shows up in a known ALS gene is likely going to be, attributed. But there are many regions of the genome that we're not looking at that cause implications for the genes. So up till now, the way that we've looked at the genome is to actually look at those pieces of the genes that get stitched back together and focused very highly on those different regions, but looking at almost nothing in between those regions. But those regions can cause splicing differences. And so now we're sequencing everybody by whole genome sequencing, which I think will yield some additional genes.
All right. Thank you so much, Dr. Harms.
Absolutely.